our attendees to join before we start. Again, everyone, we will give another minute or so for all of our attendees to join before we start. All right, thank you for joining us today. My name is Sinan, and I'm a senior bioinformatics scientist at Seven Bridges. In our webinar today, we're very excited to tell you about SARS-CoV-2 variant identification with DDL's proven Athena pipeline on the Seven Bridges platform. We will start with a brief introduction of Seven Bridges, then I will leave the floor to Dimitri and Michael from DDL, and they will take us through the Athena pipeline and its application to SARS-CoV-2 variant calling. Throughout the webinar, please feel free to ask any questions through the chat section, and we will answer as many as we can at the end. Next slide, please. So let's start with who we are. Seven Bridges is a bioinformatics solutions company that has been around over 10 years, mainly working with top pharmaceutical, biotechnology, and diagnostic companies, large public programs, as well as national initiatives. Our goal is to make research easier and faster to advance human health. And we offer a complete bioinformatics ecosystem to achieve these aims for researchers from all ends of the spectrum, ranging from bench scientists aiming to do basic research to advanced bioinformatics scientists working in pharmaceutical companies doing drug development. And by this way, we offer a holistic approach to bioinformatics. And at the core of our bioinformatics ecosystem, the Seven Bridges platform serves as the foundation, enabling those distributed teams to collaborate in a secure, compliant, and efficient manner. Our platforms currently also offer multi-cloud capabilities, enabling researchers in those distributed teams to analyze data where it resides, eliminating the need of those expensive data storage and logistic costs, further accelerating the rate of discovery. And in addition to our platform, we also offer our expert scientific services. Our on-demand professional and consultative services are custom fit to meet our users' needs. Whether our users are looking to optimize existing workflows, require assistance with some complex workflow development, or even seeking scientific peers to help determine the best analysis to perform for their data, we provide help every step of the way. Seven Bridges also develops transformative analytical approaches. So a specific product we are excited about is Graph, which is a practical, scalable, and accurate graph-based bioinformatics solution that effectively leverages known genomic variations of interest to eliminate the reference bias in sequencing experiments. And within our platforms, researchers have easy access to all public and proprietary data sets to create, view, or run analyses and explore the raw data as well as the results that they have obtained. In addition, there are more than 400 pre-built workflows across a wide range of research areas readily supported on the platforms. Additionally, users can also port their own workflows into the platform And once those workflows are optimized, they can be automated for standardized high throughput and high volume processing on the platform. And all of this is done while maintaining the data integrity through extensive checking capabilities, as well as provenance, and ensuring the long-term reproducibility of the analyses performed through the 7Bridges ecosystem. And the depth and breadth of this 7Bridges experience in developing bioinformatics solutions has been recognized by multiple awards, noting how it is easy to perform analyses on the 7Bridges platforms. Next slide, please. By leveraging the breadth of the 7Bridges bioinformatics ecosystem, researchers can go beyond the standard workflow processing to one of two iterative analysis. And this enables them to reduce the time and effort required from hypothesis generation to a product on the market. Now, researchers can analyze both genomic and phenotypic data in a secure and collaborative cloud environment that actually co-localizes the private and public data resources with standards-compliant analytical workflows that are either developed by users or readily available on the platform. This allows users to generate hypotheses, iteratively test and define those hypotheses, and identify new biological insights and bring novel products to market. And the entire platform and the 7Bridges bioinformatics ecosystem is built with security and collaboration in mind, enabling researchers to easily control access to their data as well as their applications, share data, and perform analyses together with their collaborators. The platform also enables a wide range of users with different skill sets to work together. For instance, once those workflows established and optimized, bench scientists can use our graphical user interface to use those automated workflows to process massive amounts of data for secondary or tertiary analysis, or advanced users can use our application programming interface or automation capabilities to programmatically control any aspect of the data management as well as analyses. And by harnessing these core capabilities of the 7Bridges bioinformatics ecosystem, researchers are able to enhance their hypothesis resolution processes, identify actionable insights, and even turn failing clinical trials around. Next slide, please. As Dimitri will talk about DDL's Athena Workflow, I would like to introduce the tool and workflow management capabilities of the 7Bridges platform. As mentioned, in addition to available applications covering a wide range of bioinformatics applications, our users are able to bring their own tools and workflows easily using the common workflow language and Docker containers, enabling very easy reproducibility, portability, as well as scalability. And all of this process is streamlined with visual, intuitive, and interactive tool and workflow editors that are available on the platform, which allows our users to create those standards-compliant tools and workflows without knowing the details of the standards being implemented. You can see an example of our workflow editor on the right-hand side on the slide, showing how a developer can simply drag and drop new tools to their canvas, tie them together to create their customized workflows. And while doing that, users can lock certain parameters for their workflow or expose to the users to enable them to set the values. So these functionalities pave the way for standardized and validated workflows used in controlled environments such as clinical trials. And 7Bridges platform also pays a significant attention to reproducibility. All tools and workflows are version controlled, and every change that has been done creates a new revision, ensuring the long-term reproducibility of every analysis, even after years passed. So the platform makes this whole process very easy to allow researchers to focus on science rather than dealing with establishing a reproducible infrastructure. Next slide, please. And in addition to those defined tool and workflow executions, either by using 7Bridges tools and workflows or users' own tools and workflows, 7Bridges platform also allows researchers to perform any type of ad hoc interactive analyses through integrated Jupyter Lab and RStudio environments. This eliminates the need for time-consuming and costly data logistics operations to perform any type of interactive analysis. Data Cruncher is a fully integrated feature of the platform that again accelerates that collaborative insight generation processes. Next slide, please. And lastly, I would like to introduce our partner, DDL, a state-of-the-art laboratory specializing in virology, molecular diagnostic testing, and assay development. DDL has selected 7bridges to scale up their analyses, create a simpler, integrated data management, reporting, and next-generation sequencing analysis tool that bridges gaps across its departments. With that said, I would like to introduce Dimitrios Palakanis and Michael Weber from DDL, who will talk us through their efforts at DDL and how they have developed Athena pipeline to detect SARS-CoV-2 viral variants on the 7bridges platform.
Thank you, Sinan. Thank you for this wonderful introduction, and also thank you for the opportunity to present our work. I'm Dimitri, and today I'm joined by Michiel, our senior bioinformatician at DDL Diagnostic Laboratory. And together with Michiel, we are going to answer any questions that you may have at the end. And I'm going to present you with Athena, our variant calling pipeline that we have now migrated to 7bridges, and how you can use it to aid you in the analysis of coronavirus samples. So a brief outline of what we are going to touch upon today. I'm going to start with an introduction of who we are and what we do at DDL, then give a brief overview of Athena and how it works, then proceed to a live demo to show how you can run Athena on the 7bridges platform, and also how to analyze the results that you get out of Athena. After that, I will talk about some population statistics that we have done on-site about corona samples and the data that we have generated using Athena. And finally, I have some closing discussion points, and then we will conclude with a Q&A session. So we are a leading diagnostic and clinical trial operation service company that supports clinical and preclinical studies. We are located in the Netherlands, but we operate worldwide with processing facilities around the world. We offer a wide range of services, ranging from traditional virology assays to the latest next-generation sequencing for a very broad range of pathogens. We are involved in the complete process of new drug development programs, starting from research, the preclinical and clinical phases, and all the way to the launch of a drug. We also offer post-marketing surveillance of existing drugs and vaccines. We operate at a global level, and we are the preferred testing laboratory for several of the top 10 biopharmaceutical companies. As I mentioned earlier, we offer a wide range of services, and in this webinar, I will focus on the analysis of next-generation sequencing assays. So, from going to a biological sample to NGS data, the process starts by having a sample, then isolating the RNA or DNA molecules we are interested in, performing endpoint PCR to get the amplimers, and once we have the amplimers, we can perform fragmentation and then perform NGS amplicon sequencing. So the end result of that is a set of FASTQ files. We perform paired-end sequencing, and the end result is paired-end FASTQ files. In these files, we have short genomic sequences that we call the NGS reads, and those short genomic sequences belong to our samples. And in the FASTQ file format, every set of 4 lines is considered to be one read. So once we have our short genomic sequences, the NGS reads, then we have 2 goals. You can imagine that when we have a reference genome, for example, here you can see the reference, a small portion of the reference genome of the coronavirus. The first goal that we have is to map those reads against the reference. And by that, we mean that we try to match where a genomic sequence would fall on the reference genome. So once we achieve that and we find the correct spot, then the next thing we need to do is to find the differences between our sample and the reference genome. And those differences are what we call variants, and that process is variant calling. In the context of variant calling is how Athena came into being. Athena is a quality-based variant detection program that is focused on virology, and it was developed in-house during 2014-2015. That was the first release, and it was created due to a need of a better variant caller, because until that point we were using some commercial software that we saw could not handle insertion and deletion events as we would have liked. When we made Athena, we kept the biologists in our team in mind, and we tried to make the data as easy to use for non-bioinformaticians, because once you know your way around this data, it is really easy to interpret it and use it. We have also tried to standardize the output as much as possible, and to that extent, if you would like, and we kept reporting in mind, especially to regulatory organizations, because if you want to report this data, there should be minimal fiddling around that you need to do in order to report it. Athena is validated according to GAM5 guidelines, and we have a very rigorous validation scheme, and it is also compliant to a rule of the Food and Drug Administration in the US, the FDA. This regulation is the Title 21 CFR Part 11, which is the regulation on the keeping of electronic records and electronic signatures that the FDA proposes. Athena has successfully aided in hepatitis C treatment development, and to date, we have analyzed over 20,000 samples that we have reported back to our clients. And out of those, 8,500 samples were involved in 73 clinical trials. So this data, or most of this data, was submitted to a regulatory agent and the FDA, or the FDA. Concerning the FDA, we have strong, strict compliance to their rules. There has been some draft guidelines on the submission of next-generation sequencing data, and it is worth mentioning that the Athena default settings adhere to these guidelines. Also, we are participating on the Biocompute Framework and Biocompute Objects Initiative, an initiative organized by the FDA, whose goal is to facilitate the dissemination of knowledge regarding pipelines and the data generated by those pipelines. In that way, time and cost associated with the exchange of workflows and knowledge can be minimized, and the 7 Bridges platform facilitates that effort greatly. So, as we were growing and we had more sequencers and also started analyzing more samples, we also considered moving our infrastructure to the cloud. And in that regard, we partnered with 7 Bridges because they were very flexible to all the prerequisites that we had. The reproducibility, as Sinan also touched upon, is a given on the platform, reproducibility of results. Traceability was also very important from us. So all the way from uploading FASTQ files to the platform to getting back results, everything is traceable. Compliance to data security regulations was also very important to us, and security is baked into 7 Bridges, because you can imagine, especially in a clinical trial setting, it is very important that data remains secure at all times. And finally, technical support has been and is great. And we saw that these are really technically knowledgeable people, and that was also very encouraging to partner with them. So we moved from an old situation where we were analyzing one sample at a time with Athena on a server that was constantly running those analysis scripts. And then from going sample to sample, we moved to a new paradigm with 7 bridges, where we now can analyze multiple samples at once. It is scalable, and we are only paying costs when we are analyzing samples. And in our experience, we have seen that about 50 samples of Athena takes at most 2 hours to analyze. And that is a great boost compared to what we had before. Now I will give a brief overview of Athena and how it works. On a very, very high level, what you give as input to the workflow that is now on 7 bridges and that it is Athena, you give as input FASTQ files of paired-end sequencing reads, and you get out a zip file for every sample that you analyze. In more detail, Athena has, of course, some steps internally, and I will go through these steps briefly. So Athena starts by what we call reference selection. And what we have to do at this step is to select a reference genome against which a sample can be mapped and analyzed. For coronavirus right now, this is not specifically applicable because right now there is only the Wuhan Hu1 sequence available with this accession number. But the pipeline is configurable, and if there are more reference genomes incorporated, we can incorporate those easily into an Athena analysis. So here I'm giving an example of HBV, hepatitis B virus, where there are 9 different genotypes. And what we do is that the reads are mapped against all possible references of a species, and we use by default 10% of the reads. And then a read can only be mapped to a single reference. So the reference that is selected automatically is the reference for which most of the reads mapped. So in this case, you can see an example where for a sample, 96% of the reads were mapped to genotype F, and then that will be the reference genome against which the rest of the analysis is going to be performed. So once we have selected the reference, then we perform the first mapping. So we map the NGS reads against the reference genome. In that way, we can then calculate a coverage. And this coverage we calculate per nucleotide and per position. And in that way, we can acquire the coverage graphs. And those graphs we get on full genome level, but we also get on target level or gene level. So after we have calculated the coverage, we are then able to calculate a first consensus. And the first consensus is a genomic sequence stored actually in a FASTA file. And that genomic sequence is now more similar to our sample compared to the reference genome. So once we have that, the first consensus sequence, we then map the reads in the FASTQ files again on that first consensus sequence. And we have seen that when we do that, that increases coverage by a lot. Once we acquire a second mapping, we are then able to extract a second consensus sequence. And that second consensus sequence is really the sample sequence that you get, as I mentioned, on full genome level and also on sample level. We also have 2 ambiguity thresholds. One is 25% by default, and one that can be user-defined. So after that, we perform what we call sample composition. And we call it that because we are finding exactly what a sample is composed of. So we generate a lot of records, And in contrast to other pipelines, we do not just give the top hits. We give you back everything that is detected in those FASTQ files. So we are also generating reference records, substitution records, insertion records, deletions, and gaps. And you can imagine that since reads with different genomic events map on some positions on some regions of the genome, it can be that multiple events can be registered on a single position. So for example, for a specific sample and on a specific nucleotide position, we see that 85% of the reads mapped there are the reference nucleotide T, but we can also see that there is a deletion event at 14.5%, and there are also some that have very low read counts, but are good quality. So what we do is that we do not leave them out. They are in the dataset, and you have the option to filter them out afterwards in your analysis. All these records are stored in tab-delimited text files, which you can use for further analysis in some kind of software, or you can also even use directly in Excel. So once we have sample composition, and to round things up, we have some coverage graphs, as I mentioned, and some target statistics. So these are all our QC results. We have the variant calling results that are stored in the tab-delimited text files, as I mentioned. We have the FASTA file consensus sequences. And we also have a PDF sample report that you get per sample. So all of these files are contained in the zip file that you get per sample that I mentioned in the start. This PDF sample report contains the coverage graphs and the target statistics in a clear manner with the tables and the graphs. And it is also meant to be a file companion for all the files with the variants and the consensus sequences that you get in the zip file. So a question that we need to answer, and is probably on a lot of people's minds, is how does Athena compare to other pipelines? In that degree, we have a collaboration with the FDA and the George Washington University. And there is some ongoing research on that matter. And we have seen with some generated hepatitis C samples that Athena performs very, very closely to the variant caller that the FDA uses internally. And that is a very exciting result for us. Then we also have a benchmark of multiple pipelines, which is an ongoing effort for us. And in that benchmark, we are trying to find, so we run some samples through some pipelines. And what we are trying to do then is to find all the variants that are above 1% frequency in any of the pipelines. And what we try to do next is to find if those variants are detected by any of the other pipelines at any threshold. And we can see that out of 2,900 variants that we have in this dataset, the ones that are 1% in all of them, and then that Athena detects, are 93.4%, and that is the highest, the best performing pipeline. These are, of course, preliminary results, but we are very confident about these results, and we are working on more benchmarking. So concerning a demo and how you can run Athena on the 7Bridges platform, the first step is to upload some samples on 7Bridges, of course. So for that, the most straightforward way is to use a manifest file, and that is simply a comma-separated file, a CSV file, that you can also create in Excel or other software. And In that file, you can provide the file names that you are going to, of the samples that you want to upload, and then also give some metadata attributes. These ones are the locked ones from that 7bridges uses, and these ones need to be used explicitly. And then 7bridges also offers the opportunity to give as many as you want manually, and we have harnessed that to make all the Athena variables as metadata variables. So if you define them in this manifest file, you can run Athena with those metadata attributes, and they will be picked up automatically. So when you actually run Athena, you do not need to define any parameters for your samples. So once we have those, once we have the manifest file, then 7bridges provides the 7bridges uploader program. You log into this program with the credentials that you also use on the platform, and then you can select to upload via manifest. Once you do that, then the manifest file, you choose the manifest file that you created, and that manifest file is parsed. You can see that if no errors are reported back, then you are good to continue with the upload. And then on the platform, you can select the project, and within the project, you can also select the folder where you would like to upload your FAST2 files. And once you have selected that, then those files are ready for upload, and yeah, they are uploaded to the platform. So at this point, I will give a live demo of how you can run Athena. And this process starts by going into your apps and then selecting the Athena workflow. So the Athena pipeline lives as a workflow on the platform, and then you can hit the run button. Once you do that, you just need to select some files. And because it is defined that the input is FAST2 files, the moment you select files, you get back immediately only FAST2 files to select from. So here I have a folder where I have uploaded some files already. And then what you can do is you can select those, select Save Selection, and then those files are there to be analyzed. We don't need to define anything else, and then we can just hit the Run button, and in that way, we run Athena on 7bridges. So we are not going to wait for this task to finish. I have already performed this task, and this task is finished. So what you can do afterwards is that for this task that you have just run of this task of Athena, you have some results. And just for a second to see those results. So for those results, you can see that you get those back. So you get those back on the. So you get back those files on the when you have the results, you get them in the final report output. And then what you can do is that you can see you have one zip file per sample. And then we can analyze those samples directly on Data Cruncher, as Sinan mentioned. And Data Cruncher is a very nice commodity of 7bridges, because you can analyze. You don't need to set up any Python environments. You don't even need to have your own servers. You can just use this directly in your web browser. So here, what we need to do is that I have set up a Jupyter notebook, and I have a webinar notebook ready. You need to load in some Python dependencies, and then select the task that you have run. Once you select the task, you can extract those results in individual folders. So that's what I do here. And then you can see that inside the folder, you will get all these files that I talked about in the presentation. So once we have all these files, then we can load in the results. And once we have loaded in those results, what we can then do is that we can start querying this data for some interesting findings. So for example, I can query this data for variants that are above a certain threshold. So yes, so here you can see for variants, I'm selecting only records that are not the reference records. And then here you can see that for some of the samples, we have some substitution records. And for those substitution records, you can see that some of them are also repeating for some of the samples. So this is already some interesting data. You can also see that for one of the samples here, there is a consecutive deletion on some positions, and we register that both on each nucleotide position, but also as the haplotype deletion record, so as a single record of a deletion that starts at a certain position. So then another example of what we can do is that maybe we are interested on a specific position on the genome. So what we can do then is that we can say, for my samples, how does that position look? Give me again the high frequency records at that position. So for example, we have here the first three samples that have a reference record here. The nucleotide A remains NA. But then for the other three samples, this A turns into a G. So this is a substitution. But then something like that, how does it translate to amino acid records? We can also see that from the data. And here we have an example. This position 23403, does not exactly correspond. It is the second position in that codon, in that target. So here we can query the dataset on the previous position, the first position of the codon, which is 23402. And here again, we can see the same data, but on a different level. So D amino acid remains a D, but then for the other three samples, this D turns into a G. Finally, we also have an example where we are interested in specific variants. So maybe there are some variants in literature, or maybe some variants that we've seen in one of the experiments we've made. And then we can query for these specific variants and say, can we find those in our dataset? So for that, here we are interested in some substitutions. And then we can see, for example, for the first sample, we do not have the one that is the 3037. So this data, we can filter down a bit. We can focus on the frequencies, on the percentages. And what we can do is that we can create a smaller table where we focus on those substitutions, and we look at the percentages of those substitutions in the samples. And if we plot this data, then we get a kind of heat map in this case. And this is interesting because we can see that some of the samples have very low percentages for 4 of those substitutions, but a very high percentage for the other 3. And it is the other way around for the second cluster of samples. So these distinct clusters are interesting patterns of expressions that we see. And this may require further investigation. So this is what you can do with the Athena data. And of course, everything that I've done here is inside the Jupyter notebook, but all these operations you can also perform using Excel or any other program or programming language that you like. So to get back to the presentation, What I have done now for these 6 samples in my example, in my small example, we have done for almost 600 samples of corona that we have taken from NCBI. Those samples were from 18 different countries. And what we did is that we have tried to create some population statistics. So here you can see on the x-axis, we have different substitutions. Then on the x-axis, we have the number, on the y-axis, we have the number of samples out of 589 that had that substitution. So then if we divide one number with the other, we find a percentage, and that is the percentage that you see on top of each bar. So for example, for the first one, for C241T, almost a bit over 200 samples out of 589 had that substitution, so that translates to 35%. So then what we try to do is that we saw some that are high frequency, and we thought, can we somehow validate this data? So then, for those that are highlighted here, if we zoom onto those, we could cross-reference all of them in literature. And for some of them, it's also multiple publications. So that is already a very encouraging result that the data that is coming out of Athena is valid biological data. The same we did for deletion events. And here again, in target ORF1A, we found a consecutive deletion stretch from position 510 to position 523. And again, for this part, we saw that there is some research done at Stanford University, and there they performed some multiple sequence alignment, and they have detected exactly the same deletion region. Finally, we did exactly the same for insertion records, and for especially those two, this is a single T insertion, and this is a single A insertion. This is in target ORF1B, and this is in target N. For those, we can see that since they are single insertion events, they imply that a frame shift is happening. And this is something that is known for the coronavirus. That is something that we could, again, validate in literature. So all these results show that Athena can really show you the truth of the samples, the biological truth, on your screens. So what we offer? Concluding my presentation, in the fight against the coronavirus, we are offering a free trial subscription for researchers working on SARS-CoV-2. We offer custom pipeline design, development, validation, and hosting. So if you want to tailor Athena to your needs, or if you have any other bioinformatics project, we can tailor that to your needs and host it on 7 bridges. Then a unique point for us is reference conversion. Reference conversion does not apply to coronavirus because right now there is only one reference genome, as I mentioned, the Wuhan Hu1. But for example, for hepatitis B, where there are 9 genotypes, if you, during the reference selection step, if genotype A is selected for one sample and genotype B is selected for another sample, the variant calling results that are generated for those two samples are not comparable with each other because they originated from from comparing to a different reference. So what we have implemented as part of Athena, and that is quite unique, is that we can convert the records from one reference to another reference. And that is very powerful, especially as you can imagine in a clinical trial setting, because then the results among your samples become really comparable. Finally, we offer consultancy, also regarding data reporting. And we also can offer any other kind of bioinformatics services. And we can, as I mentioned, we can tailor those to your needs exactly. This concludes my presentation, and I would like to thank you very much for your attention. If there are any questions, we would be happy to answer them. Thank you.
Thank you very much, Dimitri. We have a few questions coming from our attendees. So I would like to start with some questions on the reference sequences. So one question is, how did you determine the set of reference sequences that you used at the first reference selection? Did you use a phylo tree or pick from certain nodes on the tree?
I am sorry. Just give me 1 second, please. Let me also. Yeah, could you please repeat the question? I'm very sorry, but it's nice if you repeat the question.
Sure. So the question is, how did you determine the set of reference sequences that you used at the first reference selection? Did you use a phylo tree and pick from certain nodes or on the tree?
Yes. So yeah, we perform.
So this is Michiel. Maybe I can add to that. For corona, there's only a single reference right now, like the major genotype WHO1, WHO number 1.
Yeah.
For other pathogens, we have good connections to the virology community, and we include main genotypes by default in Athena. But it has happened before that the mapping was really bad, so the number of reads that stuck on the reference sequences wasn't really optimal. And we actually detected a new subtype by our Athena analysis. So this is usually indeed when we go and look into literature and see if other clades or genotypes have been described, and we will add them accordingly. But in general, we only use curated NCBI sequences to do this.
Thank you very much, Michiel. One other question is, do you think that observing amino acid change from only one variant can be wrong due to potential existence of upstream frame shift variants on the same gene?
Due to potential existence, you said?
Yeah, so frame shifts is definitely a phenomenon that also corona is doing. Athena will not necessarily call all substitutions after frame shift event, because sometimes you're looking at false positives or mapping errors. And in case of doubt, I would generally say Athena will not call them as a variant. But of course, if it's a true frame shift event, then any downstream codon will get affected. I mean, this is the biology, of course. So the question was, if you see only one variant change, it's definitely possible that you could link this variant to one of the insertion or deletion events that is more upstream. But that really depends on the data set and how you filter that out. But in general, yeah, it could happen. But you need to look for the link as well, I guess.
Right. And one related question is coming from our audience is, can Athena handle other respiratory viruses, or which viruses does Athena work on?
Yeah, so other respiratory viruses, we we have already set up Athena, and it is working in production with HRSV, so the human respiratory syndrome virus, and with influenza. So with these ones, it already works. And in general, Athena works with CMV, HCV, HBV, influenza and HRSV I mentioned, Corona now, and I don't think I'm missing any there. And of course, if that's as I mentioned, it is really configurable. So if you have any other type of virus that you are working with, we can definitely get it, test with it, and incorporate it into the Athena workflow. We can definitely do that.
Perfect. Thanks, Dimitri. And in terms of running the Athena pipeline, what number of samples do you find it optimal for one run of Athena workflow?
Yes, that depends. Yeah, that's a tricky question because it is also the file size matters a lot. So, for example, if you have some files that that are maybe 2 gigabytes for each FAST2 file, then you couldn't run 400 of them in one task. I would say that maybe normal sized samples around 100 you could do definitely. And if you do 100 samples, I still stand by my statement that should take less than 2 hours to perform.
Great. We have one more question about the reference. So the question is, should we first do virus reference consensus analysis to build the virus reference?
Should we, sorry, can you repeat that?
Sure. Should we first do virus reference consensus analysis to build the virus reference?
Usually we take all the different genotypes into the system. So they will become separate analysis. I mean, the reads will be compared with the genotype A or B reference. So if you make one consensus out of a set of references, it is a way to go, but you are really compressing that information then into one kind of genome sequence. It could lead to a bias in variant calling. That is why we use the nearest known reference in literature for variant calling, partly because if the reference you originally try to match against is similar to your reads, the more reads will be in the analysis and the more coverage will be the result. So if there are new references coming also for coronavirus, they will get they will go into the system as separate reference sequences. And the reference selection step in Athena will take a subset of the FASTQ file and see if the one reference is nearest, closest to that sample, or the new one.
Yeah.
Thank you, Michael. There's a question coming from audience asking, any advanced analyses that are supported through Athena, for example, phylogeny, evolution, or transmission analysis?
So for phylogeny, for example, you get FASTA file consensus sequences. So those you could definitely use to make a phylogenetic tree analysis, for example. Definitely, you can use that straight out of the box.
Yeah, so this is like phylogenetic tree building is not the default output by Athena, but we could definitely support you in this more post-processing analysis. Like Athena throws a lot of data at you, and definitely we know how to handle it. It's not that bad it's actually quite consistent. But let's say the more secondary analysis, like using all the consensus sequences of a clinical trial into phylogeny, is not part of the current output. But as Dimitri showed, even with simple notebook, we can share that as well. We can really help you set that up.
Great. If there's any other questions from the audience, you can write to the chat. Otherwise, we have one more question, which is about, do you offer any support on the interpreting the results obtained via Athena pipeline?
Yes, yeah, in that degree, we are, we can also offer interpretation of results and create custom reports for you, especially in a clinical trial setting. I can imagine that is really useful. And for the rest, we also offer. I think I'm missing something here. We also offer consultancy. We can definitely make any kind of report in the end. So interpret the data, try to get as much out of it.
Yeah. Also, if you're really doing a data submission towards FDA or another regulatory institution, we have a very standardized way of reporting. We have a data management department, so we really connect this Athena data to CDISC standards, which are really in place for data submission.
Yes.
Yeah. Thank you very much. We have one last question just arrived about, Dimitri, can you give us an idea about, again, the estimated time for analyzing the data through the Athena pipeline? I believe you mentioned about around 2 hours for 100 samples.
Yes, for normal, again, I must press on that point. For normal, what is normal? But for normal-sized samples, so something that is coming out of Illumina MySec, I guess. For a sample like that, you could do 100. And I stand by that statement. It should take around 2 hours, because that's the thing. For example, the task that I run now takes about 15 minutes, but it's not like it's going linearly with the number of samples. It's it's almost inversely proportional. You still need some time to create those spot instances and get those individual command line tools running. But once you have that up and running, then it scales really well on the platform. That is one of the main strengths of 7bridges as well. That if you do 100 samples, it doesn't mean, let's say, it takes 2 hours, and if you do 400 samples, it takes 4 hours. No, it doesn't work like that. Maybe it takes 2.5 hours to do something like that. So,
so we're at the hour, and we have no questions left. So we'd like to thank all of the attendees joining us today, and we are looking forward to seeing you again in the next 7bridges webinars. Thank you.
Thank you.